Welcome back to the deep dive. If you are here, I'm guessing it's because you are looking for the absolute cheat code, the most efficient, most comprehensive way to really master the foundations of biology and chemistry.
That's right. We've basically taken an entire syllabus, a huge stack of reading, and our goal is to distill it all down into one single cohesive story.
It's a huge undertaking.
It is. It's an ambitious project.
Yeah.
I mean, we are spanning the entire scale of existence. We're going from subatomic particles, the tiny things that govern chemical reactions, all the way up,
All the way up to the entire
the
planet.
entire biosphere. And the mission isn't just to list a few hundred definitions. It's to transform that list into a logical, interconnected mental model that actually makes sense.
That's the key right there, the connections. You should think of this not as a crash course, but as, well, the ultimate study guide.
Exactly.
We aren't just going to recite jargon at you. We're providing the context. We want to show you, for instance, how the shape of a single water molecule is responsible for the structure of your cell membranes.
And how that membrane structure controls the flow of energy that then fuels your genetic inheritance. It's all linked.
So once you see how these fundamental concepts drive each level, from an atom to a cell to a whole organism, you start to realize that life is it's operating under a universal set of rules.
And we are going to distill those absolutely critical nuggets you need to understand how life works at every single level.
Yeah.
And we have to start at the very beginning.
Which is?
What even is life? How do you define it?
Okay, let's unpack this. The ultimate litmus test. You're standing in a field, you pick up a rock, and then you look at a flower. What are the, say, non-negotiable criteria that make the flower alive and the rock not?
There are 7 of them, the 7 characteristics of life. And the key is an entity has to exhibit all 7 at the same time. If it misses even one, it's fundamentally non-living.
Even if it seems active in some way.
Right, even if it seems active.
Okay, what's number 1?
Number 1 is order. Living things are not just random piles of molecules. They possess these incredibly complex, highly specific organizational structures.
Right, like a neuron has a totally different structure than a skin cell.
Exactly. Both are intricate, but they have a specialized, defined organization. The rock does not.
And that organization has to be passed on, so that must be number 2.
Reproduction.
Reproduction. Organisms have to be able to produce viable offspring.
And those offspring, they have to grow.
Which is number 3, growth and development. And this is important. It's not just random growth like a crystal getting bigger. This growth has to be directed by specific instructions.
The instructions encoded in its DNA.
Precisely.
Okay, number 4 has to be about energy, right? It takes work to maintain all that order.
Energy processing. Life requires a constant input of energy. Organisms have to perform chemical work just to stay organized, usually by taking in energy from their environment.
Okay, so you have order, you pass it on, you grow, you process energy. What's 5?
Regulation. This is where the complexity really shines.
Yeah.
Organisms have to maintain their internal conditions within a very narrow, stable range.
That's homeostasis. That's homeostasis. Think about how your body keeps its temperature at 98.6 degrees or manages your blood sugar. Without that constant regulation, the complex order just falls apart instantly.
Okay, 6. The ability to react.
Response to the environment? It's the ability to sense and react to stimuli. It could be a single-celled organism moving towards food, or you pulling your hand away from a hot stove. It's a fundamental sign of life.
And the last one. This is the big one, the one that works over huge time scales.
The 7th, and maybe the most powerful trait, evolutionary adaptation. Individual organisms respond to their environment, sure, but it's the population that changes over generations.
So advantageous traits get passed on more often.
And they accumulate, making the population as a whole better suited to its environment. That's natural selection in a nutshell. It's those 7 traits: order, reproduction, growth, energy, regulation, response, and adaptation, all working together that define what it means to be alive.
That context is so important. You can have a crystal that has order and it grows by adding more particles, but it completely fails on regulation, on energy processing, on DNA-directed development. It's not alive.
Not even close.
So if an entity passes this 7-point litmus test, we can say the basic functional unit of that life is the cell.
Absolutely. And that's formalized in what we call the cellular theory. It has 3 crucial parts.
Okay.
1, all living things are made of 1 or more cells.
Yeah.
2, the cell is the basic fundamental unit of all life. And 3, all cells can only arise from pre-existing cells.
That last one is huge. It completely refutes the old idea of spontaneous generation, that life could just pop out of nowhere.
Right. No, cells have to come from other cells.
So even though there's this massive diversity of cells out there, they all have to share some core features, right? Some non-negotiable architecture.
They do. There are 4 universal features. Every single cell, no matter what, has to have a plasma membrane.
The border, the
Keep it.
gatekeeper.
The gatekeepers, yeah. It creates an internal world separate from the outside. Second, it has to contain DNA.
The instruction manual.
Instruction manual. Third, the inside is filled with cytoplasm, which is the internal fluid matrix. And finally, they all have to have ribosomes.
Which are the little machines that build proteins.
Exactly, the protein factories. Without those 4 things, membrane, DNA, cytoplasm, and ribosomes, you don't have a cell, period.
And this brings us to probably the most crucial distinction in all of biology. The 2 fundamental types of cells that divide all known life.
Right. We contrast the prokaryotic cells with the eukaryotic cells.
Let's start with the prokaryotes. These are the older models, right?
The originals, yeah.
Mhm.
Think bacteria and archaea. They're the evolutionarily older, simpler forms. They're smaller. They generally lack complex internal compartments. And the big defining feature,
No nucleus.
they have no nucleus. Their DNA is just found freely floating in the cytoplasm.
Okay, so then you have the eukaryotes. That's us. Animals, plants, fungi.
Right. These are the highly organized, much more complex cells. They're larger. They contain all these specialized membrane-bound compartments that we call organelles.
And the big difference, the DNA is safely tucked away.
It's housed within a dedicated nucleus. This organizational leap, put in the control center in its own protected room, was a massive evolutionary jump.
So that jump from a simple prokaryote to a complex eukaryote required a huge increase in organization. And that structure just keeps building up, doesn't it, through the biological hierarchy.
It does. We go from the smallest chemical units all the way up to the entire globe.
Let's trace that journey. We have to start at the absolute smallest level, the chemical unit, the molecule.
Right. And molecules combine and specialize to form organelles,
The tiny organs inside the cell.
which are inside the cell, the basic unit of life.
Then when you scale up from there, you get cells working together.
Similar cells specialize and cooperate to form a tissue. Then you get different tissues working together to create an organ, like the heart or the liver.
And multiple organs team up for a major purpose, like digestion. That's an organ system.
And all those systems integrated together form the individual organism.
Okay, so we've built a single living thing. Now we have to zoom out and look at how they interact.
This is where ecology begins. A group of organisms of the same species in the same area, that's a population.
And if you take all the different populations in that area, every living thing, from the bacteria in the soil to the trees to the bears, what's that?
That's the community.
But the community doesn't exist in a vacuum. It needs the non-living stuff, too.
Exactly. The ecosystem level adds in those crucial non-living or abiotic elements, the soil, the water, the sunlight, the air. It's the community plus its physical environment.
And bigger than that.
Multiple ecosystems interacting across a region form a landscape. And the absolute pinnacle, the sum total of all life and every ecosystem on Earth, is the biosphere.
It's a structure that really shows that idea of emergent properties, right?
Yeah.
At each new level of the hierarchy, new, more complex characteristics appear that just weren't there in the level below it.
That's the perfect way to put it. The whole is truly greater than the sum of its parts.
Understanding that incredible hierarchy, all that complexity, it just begs the question, how do we know all this stuff? We have to turn to the tool we use to generate reliable knowledge, the structure of science itself.
We rely on the scientific method. And it's so much more than guesswork. It's a self-correcting, systematic approach to understanding the world.
And for anyone studying, knowing the steps is key. There's a formal process, usually broken down into 6 steps. It all starts with just looking around.
Careful observation. You notice a phenomenon, which leads you to step 2, a focused question. Why does this happen? Or how does that work?
Step 3 is where the real science begins. You form a hypothesis.
And a hypothesis is more than just a guess. It has to be a proposed, testable explanation. And critically, it has to be falsifiable.
Meaning you have to be able to prove it wrong.
Exactly. That falsifiability is the core strength of the whole method. You have to be willing to be wrong.
Okay, so you have your testable hypothesis. Next, you run the experiment to see if your prediction holds up. Then, after collecting all the data, you move to the analysis phase.
Right, often involving statistics to see if your results are significant. And finally, you draw your conclusion. The data forces you to either accept or, well, support your hypothesis or to reject it.
And it's important to note, even if the data supports it, scientists don't usually say they've proven it right.
That's a great point. We say the evidence supports the hypothesis. It leaves the door open for future experiments and future refinement. Science is always provisional.
And when you're setting up that experiment, you have to be really strict about controlling things. The one factor you, the experimenter, intentionally change is.
The independent variable
And the thing you measure to see the effect of that change.
is the dependent variable. Getting that relationship crystal clear is, it's half the battle in experimental design.
We also get two different kinds of data from these experiments.
We do. We get qualitative data, which is descriptive. The solution turned blue or the culture looks cloudy.
And the other kind is what we usually prefer in science.
Quantitative data. That's the numerical data, measuring the exact cell count or the precise temperature change. It's preferred because it allows for much more powerful statistical analysis.
And underlying this whole method are two major ways of thinking, of using logic. What's the difference between inductive and deductive reasoning?
Inductive reasoning is about moving from the specific to the general. It's about pattern recognition.
So if I observe that every dog, cat, and monkey I see is warm-blooded,
You might induce the general principle that all mammals are warm-blooded. It's great for generating broad generalizations and new hypotheses.
okay, so what's deductive reasoning?
Deductive reasoning is the inverse. It's the backbone of hypothesis testing. You start with a general principle like all living things are made of cells, and you use it to make a specific testable prediction.
Like, if I look at this new organism under a microscope, I predict I will see cells.
Exactly. And if your specific prediction holds true in the experiment, it strengthens your confidence in that general principle.
So it's this rigorous, self-correcting process that has led us to the most unifying, powerful explanation for the sheer diversity of life on Earth: evolution.
That's right.
Which takes us straight to Darwin. His theory of natural selection wasn't some sudden flash of genius out of nowhere. It was a powerful set of inferences built on two very careful observations.
And these were observations that were pretty much undeniable, even in his time. Observation 1. Populations vary in their inherited traits. Just look around. There's immense variety, different colors, sizes, speeds, and we know those variations are passed down.
And observation 2 is about competition.
Right. Organisms produce more offspring than the environment can possibly support. This inevitably leads to a struggle for existence, a competition for limited resources.
So you have variation and you have competition. That led Darwin to his two crucial inferences.
Inference 1. In that struggle for existence, individuals whose inherited traits give them some kind of advantage, what he called favorable traits, they tend to survive better and reproduce more successfully. They are a better fit for their specific environment.
And because those successful individuals reproduce more,
Inference 2. Those advantageous traits are inherited, so they will inevitably accumulate in the population over generations. This non-random differential survival, that's the mechanism. That is natural selection. The environment itself is doing the selecting.
which means the definition of fitness is totally dependent on the context. A trait that's great today might be a huge liability tomorrow if the environment changes.
Absolutely.
Now, we should contrast that with artificial selection.
Right. Artificial selection uses the exact same principle of differential reproduction, but the selection criteria are chosen by humans, not by nature.
Like when we breed crops for disease resistance or dogs for specific temperaments.
Yeah, exactly, with the selective pressure in that case.
And through this selection, organisms find their place, their specialized ecological roles. The big ones to know are the producers and the decomposers.
Right. The producers are usually plants or algae. They're the autotrophs that make their own food, typically through photosynthesis, and they form the base of almost every food chain.
And just as important, but on the other end of the cycle, you have the decomposers.
The great recyclers. Bacteria and fungi that break down dead organic matter, returning all those essential nutrients back into the ecosystem so the producers can use them again. They keep the whole system from grinding to a halt.
Okay, we've established the logic of life, the hierarchy, and the mechanism of change. Now we have to zoom way, way down. We have to get to the subatomic level because everything we've talked about, from how a cell gets energy to how DNA is structured, is all fundamentally governed by chemistry. It all starts with matter.
And matter is just anything that has mass and takes up space. The purest form of matter is an element, which is a substance that can't be broken down chemically. It's made of only one type of atom.
And we should also mention trace elements.
Right. These are elements that are absolutely essential for life, but you only need them in tiny, tiny amounts, like iron for your blood or iodine for your thyroid.
So the smallest unit of any element is the atom. Let's do a quick tour of its internal structure.
Okay, so the center, the dense core, is the atomic nucleus. This is where the heavy particles are. You've got your protons, which have a positive charge and define what the element is. And you have the neutrons, which are neutral.
And they both have a mass of about 1 atomic mass unit, or a mu.
Roughly, yes. Surrounding that dense nucleus is the vast, mostly empty space called the electron cloud.
This is where the action happens.
And this is where all the chemistry happens. The electrons are in this cloud. They have a negative charge and almost no mass. But here's the critical part. It's the arrangement and number of the outermost electrons that determines how an atom will react chemically.
And there are a few important variations of atoms we need to define. What's an isotope?
An isotope is an atom of an element that has a different number of neutrons. The number of protons is the same, so it's still the same element, it just has a different mass.
And some of those are unstable, right?
Right. Most are stable, but some are not, which leads to radioactivity, the spontaneous emission of energy from an unstable nucleus.
Okay, and what about an ion?
An ion is an atom that has gained or lost 1 or more electrons. This gives it a net electrical charge, either positive or negative.
So the universal goal for atoms is to be stable, which they usually achieve by filling up their outermost electron shells. And they do that by interacting with other atoms, forming chemical bonds. Let's talk about the 2 big ones.
The first type is ionic bonding. This is what happens when one atom is so much more electronegative than another that it just completely strips an electron away from it.
It's a transfer, not a sharing.
A total transfer. This creates 2 oppositely charged ions, which are then held together by a powerful electrostatic attraction. A great example is table salt sodium chloride.
But the real workhorse of biology is the other type of bond.
Covalent bonding. This is all about sharing electrons between 2 atoms. Now, when that sharing is perfectly equal, you get a nonpolar molecule. But here's where the physics of life really begins. Often, the sharing is unequal.
And that unequal sharing is because of electronegativity.
Exactly. Electronegativity is just an atom's inherent pull on shared electrons. Oxygen is a classic bully. It's highly electronegative.
So when it bonds with something like hydrogen,
It pulls those shared electrons much closer to itself. This makes the oxygen end of the molecule slightly negative and the hydrogen end slightly positive. It creates a polar molecule with these partial charges.
and polarity is the secret sauce for life.
It absolutely is, because it allows for the formation of hydrogen bonds.
Now, these aren't true bonds, right? They're more like weak attractions.
That's a perfect description. They're weak temporary attractions between the partial positive charge on a hydrogen in one polar molecule and the partial negative charge on an oxygen or nitrogen in a nearby molecule.
So why are these weak forces so important?
Because their weakness is their strength. They're weak enough to form and break easily, which allows DNA strands to unzip for replication or enzymes to change shape. But collectively, when you have millions of them, they're strong enough to hold together massive structures like the DNA double helix or the 3D shape of a protein.
And there is no better example of the power of hydrogen bonds than in water. It's the solvent of life for a reason.
All of water's unique life-giving properties stem from its polarity and its ability to constantly form and reform hydrogen bonds with itself. We see cohesion, where water molecules stick tightly to other water molecules.
Which creates high surface tension. That's why some insects can literally walk on water.
Right. And we also see adhesion, where water molecules stick to other polar surfaces. This is how water climbs up the inside of a plant stem, fighting gravity.
It also acts as a great temperature buffer.
A phenomenal one. Its ability to absorb a lot of heat without changing its own temperature much helps maintain homeostasis, both inside a cell and for the entire planet's climate.
So when we mix things in water, we create a solution. Let's define the parts.
A solution is a homogeneous mixture. The substance that gets dissolved is the solute, and the substance doing the dissolving, in this case water, is the solvent.
And water is often called the universal solvent.
It is, because its polarity allows it to dissolve so many other polar and ionic compounds. It's fantastic at pulling things apart.
Okay, finally, water itself can slightly break apart into ions. This gives us the pH scale.
Right. A water molecule can reversibly dissociate into a hydrogen ion, Hu, and a hydroxide ion, OH. The pH scale, which runs from 0 to 14, is just a measure of the concentration of those free Hu ions in a solution.
So a substance that increases the Hu concentration is an acid.
It's an acid and its pH will be below 7. The lower the number, the more acidic it is.
And a substance that decreases the HU concentration is a base.
It's a base, and its pH will be above 7. And it's really important to remember the scale is logarithmic. A pH of 3 is actually 10 times more acidic than a pH of 4.
This balance is constantly shifting in all the chemical reactions of life, where we see reactants get converted into products.
It's constant dance.
So we have built the atomic and the chemical bond foundation. Now we get to use that chemistry to build the actual structures of life, the 4 massive biological molecules, the macromolecules.
We're now deep in the world of organic chemistry, which is the study of compounds that contain both carbon and hydrogen.
Carbon is the star of the show.
It's the backbone of life. Its ability to form 4 stable covalent bonds allows for an incredible diversity of structures. You even see this in isomers molecules that have the exact same chemical formula but different structural arrangements, which gives them totally different functions.
And all 4 classes of these macromolecules are built using the same fundamental design principle. You have small building blocks that you link together to form these massive chains.
Polymers. It's like building with Legos. You have a few types of bricks, the monomers, and you can build almost anything, the polymers.
And the cell has 2 beautifully simple opposite reactions to manage all this construction.
It does. To build a polymer, to link those monomers together, the cell performs a dehydration reaction. It removes a molecule of water to form the bond.
And to break it down,
It does the opposite, a hydrolysis reaction. The name literally means breaking with water. The cell adds a water molecule back, and that splits the bond, releasing the monomers.
Okay, let's get to the first class. Carbohydrates. These are the sugars. They're all about short-term energy and structure.
Right. Their monomer is the monosaccharide, or a simple sugar. Glucose is the most famous one.
And when you link a bunch of those together, you get a polysaccharide.
The polysaccharides have 2 main jobs. First is energy storage. In plants, they store it as starch. For us, that's easy to digest.
And in animals.
We store glucose as glycogen, mostly in our liver and muscles, ready for quick energy release when we need it.
And the second job is structure.
Right. For structure, plants use cellulose to build their rigid cell walls. That's dietary fiber for us. It's tough to break down. And things like fungi and insects use a similar polysaccharide called chitin for their cell walls and exoskeletons.
Okay, class number 2. Lipids. The fats and oils.
These are a bit different because they aren't defined by a true monomer-polymer structure. They're defined by a property. They are hydrophobic macromolecules. They do not mix with water.
And their jobs are long-term energy storage, insulation, and membranes.
And membranes, which is a huge one. When we look at fats, we can classify them. A saturated fatty acid has no double bonds in its carbon chain.
It's saturated with hydrogens.
Exactly. This makes the chain straight so they can pack together very tightly, which is why they are solid at room temperature, like butter.
An unsaturated fatty acid, on the other hand, has at least one double bond.
And that double bond creates a kink, or a bend, in the chain. This prevents them from packing tightly together, which is why they are liquid at room temperature, like olive oil.
We should also quickly mention trans fat.
We should. This is an artificial fat created by adding hydrogen back to unsaturated fats. It creates a molecular structure that is particularly bad for cardiovascular health.
But the most important lipid for life is the phospholipid.
Absolutely. This molecule is the foundation of every single cell membrane, and that's because it's amphipathic.
Meaning it has 2 opposing personalities?
It does. It has a hydrophilic head, which is polar and loves water, and 2 long hydrophobic tails, which are nonpolar and hate water.
So what happens when you throw these into water?
They spontaneously self-assemble into a lipid belayer. It's a double-layered sheet where all the hydrophilic heads face outward towards the water inside and outside the cell, and all the hydrophobic tails tuck into the middle away from the water.
This spontaneous organization is the basis of the cell membrane. It just happens.
It just happens, driven by those hydrophobic interactions. They can also form little spheres called liposomes or micelles.
We also have steroids in the lipid category.
Right. They are lipids with a distinct structure of 4 fused carbon rings. They can be structural, like cholesterol in our membranes, or act as signals, like hormones.
Okay, macromolecule number 3. Proteins. These are the workhorses.
Arguably the most functionally diverse class. They do almost everything in the cell. Their monomer is the amino acid, and they are linked together by strong peptide bonds.
And a major function is acting as enzymes.
The biological catalysts that speed up virtually all chemical reactions.
A protein's function is completely dictated by its 3D shape. So we have to understand the 4 levels of its structure. It's folding.
We do. The primary structure is just the linear sequence of amino acids. It's this specific order, like letters in a word.
Then that chain starts to fold.
Right. The secondary structure consists of local, repetitive folding patterns. These are caused by hydrogen bonds forming between atoms in the polypeptide backbone, creating shapes like a spiral called the I helix, or a folded sheet called the O sheet.
And then the whole thing folds up into a final shape.
That's the tertiary structure. It's the overall, final 3D shape of a single polypeptide chain. This is driven by all sorts of interactions between the amino acid side chains.
And a key driver of this is hydrophobic collapse.
That's the one. The nonpolar hydrophobic side chains all try to get away from the water, so they cluster together in the protein's core, which forces the whole thing to fold into a stable, functional shape.
And some proteins need a final level of organization.
Some do. The quaternary structure only exists if a functional protein is made of 2 or more separate polypeptide chains that have to assemble together. Hemoglobin in your blood is a classic example. It's made of 4 chains.
And if that intricate structure is lost, if the protein unfolds, a process called denaturation, it loses its function.
Completely. Which is why maintaining homeostasis, stable temperature, and pH is so critical for life.
Okay, our final class of macromolecules, the nucleic acids, the information system.
This is the blueprint in the messenger service. The monomer here is the nucleotide.
And a nucleotide has 3 parts.
It does. A nitrogenous base, that's A, T, C, G, or U, a 5-carbon sugar, and a phosphate group.
And we have to contrast the 2 main types, DNA versus RNA.
Right. DNA, or deoxyribonucleic acid, is almost always double-stranded. It uses the sugar deoxyribose. And its one essential function is to store genetic information for the long term.
And RNA, ribonucleic acid.
RNA is usually single-stranded. It uses the sugar ribose. And its job is much more active to transmit information and help actually build the proteins. It's the go-between.
And they both follow a key rule of pairing up.
Chargaff's rules, or complementary base pairing. Adenine, A, always pairs with thymine, T, in DNA, or uracil, U, in RNA. And guanine, G, always pairs with cytosine, C.
And in DNA, this pairing creates the famous double helix shape, where the 2 strands run in opposite directions. They're anti-parallel.
Correct. One runs up and the other runs down. It's a beautiful, stable structure for storing the code of life.
Okay, we have the building blocks. Let's assemble the city. We're now taking a tour of the eukaryotic cell. But before we even go inside, we have to talk about the physical law that limits how big any cell can get.
That's the volume to surface area ratio. This is a huge concept. As a cell gets bigger, its internal volume increases much faster than its outer surface area, the plasma membrane.
And the cell needs that surface area to exchange everything it needs, nutrients in, waste out.
Exactly. If the volume gets too large, the surface area just can't keep up with the demand. The cell would starve or poison itself. This is why cells have to be small and why large organisms have to be multicellular.
All right, so let's step past that plasma membrane into the internal fluid, the cytosol or cytoplasm. Dead center in our eukaryotic cell is the command center.
The nucleus. It's a double-membraned organelle that houses and protects all of the cell's DNA.
And the instructions from that DNA are used to build proteins, which happens on the ribosomes.
Right. Ribosomes can be free-floating in the cytosol, or they can be attached to the rough endoplasmic reticulum, the RER.
Why is it rough?
Because it's studded with ribosomes, which gives it a bumpy look. The RER's job is to modify proteins that are going to be secreted from the cell or embedded in a membrane.
And its partner is the smooth endoplasmic reticulum, or SER.
Which is smooth because it lacks ribosomes. Its job is different. It's specialized for things like lipid synthesis, detoxifying drugs and poisons in the liver, and storing calcium ions.
From the ER, where do the newly made proteins go?
They travel to the Golgi apparatus. You can think of this as the cell's post office or finishing and shipping center.
So it modifies, sorts, and packages them.
Precisely. It packages them into little membrane sacs called vesicles and sends them to their final destinations, either inside or outside the cell.
What about waste disposal and recycling?
For that, you have the lysosome. It's basically a sac of powerful digestive enzymes that can break down old organelles, food particles, anything the cell needs to get rid of.
And for storage.
That's the job of the vacuole. It stores water, nutrients, pigments, and waste. In plant cells, they have a huge central vacuole that can take up most of the cell's volume.
There's also the peroxisome. What does that do?
The peroxisome is a specialist. It's involved in breaking down fatty acids and detoxifying certain harmful compounds, producing hydrogen peroxide as a byproduct, which it then safely breaks down.
Okay, let's talk about energy, the power plants of the cell.
There too, the mitochondria are the sites of cellular respiration. They take fuel, like glucose, and convert it into the cell's main energy currency, ATP.
And in plants and algae, there's another one.
The chloroplasts. These are the sites of photosynthesis, where light energy is captured and converted into chemical energy in the form of sugar.
And both of these organelles are thought to have a pretty wild origin story, right? The endosymbiotic theory.
It's an amazing story. The theory is that both mitochondria and chloroplasts were once free-living prokaryotic cells that were engulfed by a larger host cell and formed a symbiotic relationship, eventually becoming permanent organelles.
What about the cell's internal skeleton, its shape and structure?
That's the job of the cytoskeleton. It's an intricate network of protein fibers that provide structural support, anchors organelles, and also acts as a highway system for transport within the cell.
And it can also be involved in movement?
Yes. Many cells have cilia, which are short, numerous hair-like projections, or flagella, which are long, whip-like tails, both used for propulsion.
And we should mention that plant cells have an extra layer of protection outside their membrane.
They do. A rigid cell wall made primarily of cellulose, which gives them their fixed shape and structural support.
Okay, since that membrane is the border, we need to talk about how things get across it. We mentioned the fluid mosaic model.
Right. It's a fluid bilayer of phospholipids with a mosaic of different proteins embedded within it. And its key property is selective permeability. It controls what gets in and out.
And movement can be divided into two main categories based on energy use.
Exactly. First, you have passive transport, which requires absolutely no energy from the cell. It relies on the natural tendency of molecules to spread out.
That's diffusion, moving down a concentration gradient from an area of high concentration to low concentration.
Right. And if the molecule is small and nonpolar, like oxygen or carbon dioxide, it can slip right through the lipid bilayer. We call that simple diffusion.
But what about things that are larger or charged, like ions and polar molecules? They can't just slide through.
They can't. They need help. They use facilitated diffusion. They still move down their concentration gradient, so it's still passive, but they move through a specific channel or carrier protein embedded in the membrane.
A great example would be the aquaporins.
Perfect example.
Yeah.
These are channel proteins specifically designed for the rapid transport of water across the membrane.
And the movement of water itself is so important it gets its own name.
Osmosis. It's the diffusion of water across a selectively permeable membrane. And the key thing to remember is that water always moves toward the area of higher solute concentration.
The consequences of this are huge for cells.
They are. If you place a cell in a hypotonic solution 1 with a lower solute concentration, then the cell's interior water will rush in and the cell could swell and burst.
And the opposite.
A hypertonic solution, one with a higher solute concentration, water will rush out of the cell, causing it to shrivel and shrink.
So the ideal state for an animal cell is an isotonic solution, where the concentrations are equal and there's no net movement of water.
The sweet spot.
But what if a cell needs to move something against its concentration gradient, from low concentration to high?
For that, you need energy. This is active transport. It's an uphill battle, so the cell has to spend ATP to do it.
The classic example of this is the sodium-potassium pump.
It is. It uses ATP to constantly pump 3 sodium ions out of the cell for every 2 potassium ions it pumps in. This is incredibly important for maintaining the electrochemical gradients needed for nerve function. It's a huge energy expense for the body.
What about really big stuff, stuff that's too large even for a protein pump?
For that, the cell uses bulk transport. Exocytosis is the process of moving things out. An internal vesicle fuses with the plasma membrane and releases its contents to the outside.
And the reverse is endocytosis.
Moving things in. The membrane folds inward to engulf a substance, pinching off to form a new vesicle inside the cell.
All of this movement, all this structure, it all requires power. So now we have to get into the rules of metabolism, how cells manage energy.
Right. Energy comes in 2 main states. Kinetic energy, which is the energy of motion, and potential energy, which is stored energy, often locked away in chemical bonds.
And the first most important rule is the first law of thermodynamics.
Energy is conserved. It cannot be created or destroyed, only transferred or transformed from one form to another.
And chemical reactions are classified by how they handle that energy.
An exergonic reaction is one that releases energy. The products have less potential energy than the reactants did, Think of it as a downhill reaction.
And an endergonic reaction.
That's an uphill reaction. It requires an input of energy to proceed. The products have more potential energy than the reactants.
And metabolism is just the sum of all these reactions in an organism.
All of them. And we can divide metabolism into two types of pathways. Catabolic pathways are the ones that break down complex molecules and release energy. They are exergonic.
Like breaking down glucose.
Exactly. And anabolic pathways are the ones that build complex molecules and consume energy. They're endergonic.
And the main energy currency that links these two is ATP.
ATP, adenosine triphosphate, is the cell's rechargeable battery. The cell performs energy coupling, using the energy released by catabolic reactions to recharge its ATP, and then using that charged ATP to power its anabolic reactions.
But these reactions wouldn't happen nearly fast enough on their own. They need a catalyst.
They need enzymes. Enzymes are proteins that act as biological catalysts. They dramatically speed up reactions without being consumed in the process.
And they do this by lowering the activation energy.
Right. The activation energy is that initial energy barrier that you have to overcome to get a reaction started. Enzymes lower that barrier, making it much easier for the reaction to happen.
How do they do that?
An enzyme has a specific region called the active site, which is shaped to bind to its specific target molecule, the substrate.
It's not a rigid lock and key fit, though?
No, it's more dynamic. It follows the induced fit model. When the substrate binds, the enzyme changes its shape slightly to grip it more tightly, stressing its bonds and pushing it toward the transition state.
And sometimes enzymes need a little help.
They do. They sometimes require non-protein helpers called cofactors, which can be metal ions or organic molecules called coenzymes, like many vitamins.
Okay, let's look at the centerpiece of catabolism, cellular respiration. This is how we get the maximum energy out of glucose.
The overall goal is simple, but the process has a lot of moving parts. The big picture equation is 1 glucose plus 6 oxygen yields 6 carbon dioxide, 6 water, and a whole lot of ATP.
And at its heart, this is a redox reaction.
It is. It's all about transferring electrons. You just have to remember, oxidation is loss of electrons, and reduction is gain of electrons. Oil rig.
And special carrier molecules like NADU and FAD shuttle these high-energy electrons through the process.
They act as the electron taxis. They pick up electrons from glucose and drop them off later in the process.
So the whole thing starts in the cytoplasm with phase 1, glycolysis.
Glycolysis literally means sugar splitting. It takes 1 molecule of glucose and breaks it in half, forming 2 molecules of pyruvate. This process actually nets you a small profit of 2 ATP and 2 NADH, and importantly, it doesn't require oxygen.
After glycolysis, if oxygen is present, the pyruvate moves into the mitochondria for phase 2.
Pyruvate oxidation. This is a quick step that converts the 2 pyruvate molecules into 2 molecules of acetyl-CoA, releasing some co-uros and making more NADH.
Now we're ready for phase 3, the citric acid cycle, also known as the Krebs cycle.
Right, this happens in the mitochondrial matrix. The acetyl-CoA is fed into a circular series of reactions that finishes the job of oxidizing the original glucose molecule, releasing all the remaining carbon atoms as ET.
And its main output isn't ATP, right?
No, it only makes a little bit of ATP directly. Its main job is to load up a huge number of those electron taxis, NADH and FADHs.
Because all those loaded taxis are headed for the grand finale, phase 4.
Oxidative phosphorylation. This is where the massive ATP payoff happens, and it has 2 parts. First is the electron transport chain, or ETC.
What happens there?
All the NADH and FADHs drop off their high-energy electrons at a series of protein complexes embedded in the inner mitochondrial membrane. As the electrons are passed down the chain like a bucket brigade, the energy they release is used to pump AT ions into the space between the 2 mitochondrial membranes.
So you're building up a huge gradient of protons.
A massive electrochemical gradient. That's the potential energy, like water held back by a dam.
And the second part, chemiosmosis, is how you cash in on that energy.
Exactly. The halions want to rush back down their gradient, but the only path they can take is through a remarkable protein channel called ATP synthase.
And as the protons flow through it,
It spins like a tiny molecular turbine, and that spinning motion physically mashes ADP and phosphate together to synthesize huge quantities of ATP.
so what is the role of oxygen in all of this?
Its role is absolutely critical. It's a final electron acceptor. At the very end of the electron transport chain, oxygen is waiting to accept those spent low-energy electrons. Without oxygen to clear them away, the whole chain would back up and grind to a halt.
And when it accepts the electrons, it combines with protons to form water.
To form water, HEO, that's why we breathe oxygen.
But what if there's no oxygen available?
Then the cell has to resort to fermentation. It's a way to generate a small amount of ATP just from glycolysis alone by recycling the NADO needed to keep it running.
And we see two main types.
In our muscle cells, during intense exercise, we do lactic acid fermentation, which produces lactate. In yeast, they do alcoholic fermentation, producing ethanol and co-uro, which is how we get bread and beer.
Okay, that was the big catabolic pathway. Now for the primary anabolic pathway, photosynthesis. This is how energy gets into the ecosystem in the first place.
Right. This is the process that captures light energy from the sun and converts it into chemical energy stored in organic molecules.
And the overall equation is basically the reverse of respiration.
It is. 6 co-uro plus 6 uro plus light energy yields 1 glucose plus 6 oxygen. And because it requires an input of energy, it's an endergonic process.
This all happens inside the chloroplasts, and it's also a redox reaction, but the roles are swapped.
They are. In photosynthesis, HEO is oxidized, it loses electrons, and CoVO is reduced, it gains electrons. And the pigment that absorbs the light energy to get this all started is chlorophyll.
Which reflects green light, and that's why plants look green.
Exactly.
So the process has two major phases. Let's start with phase 1.
The light reactions. These happen in the stacks of internal membranes inside the chloroplast, called the thylakoids.
And what's the goal here?
The goal is to absorb light energy and use it to do two things: generate ATP and load up an electron carrier called NADPH. The process starts when light hits photosystem 2, which gets so excited it splits a water molecule to get electrons, releasing oxygen gas as a byproduct.
So the oxygen we breathe comes from the splitting of water in photosynthesis.
It does. Those electrons then get passed to photosystem I and eventually used to create NADPH. And the process also generates ATP using a chemiosmosis system, very similar to the one in mitochondria.
Okay, so you've made ATP and NADPH. Now for phase 2.
Phase 2 is the Calvin cycle. This happens in the stroma, which is the fluid surrounding the thylakoids.
And this is where the sugar is actually built.
This is where the synthesis happens. The cycle uses the ATP and NADPH from the light reactions as fuel and electrons to power a process called carbon fixation. It takes CO2 from the atmosphere and incorporates it into an existing organic molecule.
And the final output.
The final output of the cycle is a 3-carbon sugar called G3P. The plant can then use that G3P to build glucose for energy, starch for storage, or cellulose to build its cell walls.
All right, we've covered structure and energy flow. Now we have to talk about the master instructions, the genetic code, how it's copied, how it's passed on, and how it's controlled. This all starts with how DNA is managed in the nucleus.
DNA can exist in different states. Most of the time, when the cell is just doing its job, the DNA is uncoiled and accessible. We call that chromatin.
But when the cell is getting ready to divide,
It condenses that chromatin into tight, dense, visible packages called chromosomes. This makes it much easier to move around without getting tangled.
and after the DNA has been copied, each chromosome is made of 2 identical copies, right?
Right. They're called sister chromatids, and they're joined together at the center. And we inherit our DNA in homologous chromosome pairs, 1 chromosome from our mother and 1 from our father for each pair.
So most of a cell's life is spent in interphase, which is divided into G, S, and G phases. The S phase is where DNA replication happens.
Exactly. Then the cell is ready to divide. And the main type of division for growth and repair is mitosis.
The goal of mitosis is to produce 2 genetically identical daughter cells, or somatic cells.
Right. And it has distinct stages. You have prophase, prometaphase, then metaphase, where the chromosomes all align neatly at the center of the cell.
Then anaphase, where the sister chromatids are pulled apart to opposite poles.
Followed by telophase, and finally cytokinesis, which is the actual division of the cytoplasm into 2 separate cells.
And when this process goes wrong, when the cell divides uncontrollably,
That's the basis of cancer. The cells ignore the normal checkpoints and just keep dividing, forming tumors that can become malignant and metastatic, meaning they can spread to other parts of the body.
okay, so that's mitosis. The other more complex type of division is meiosis.
Right. And meiosis has a very specific purpose: to produce haploid gametes, or sex cells, for sexual reproduction.
Haploid, meaning they have half the number of chromosomes.
Half the number. So that when 2 gametes fuse during fertilization, the resulting offspring has the correct full set of chromosomes.
And meiosis is the engine of genetic variation.
It absolutely is. 2 key events create this variation. First, in meiosis of the line, you have crossing over, where the homologous chromosomes literally swap segments of DNA with each other, creating new combinations of genes.
And the second source of variation.
Independent assortment. This is the completely random way that the homologous pairs line up at the metaphase plate. In humans, just from this random alignment alone, there are 2 to the power of 23 possible combinations. That's over 8 million.
It's this variation that Gregor Mendel was trying to explain with his pea plant experiments. He gave us the language of genetics.
He did. He defined a heritable feature as a character, like flower color, and a specific variant of that character as a trait, like purple or white flowers.
And he distinguished between the genotype, the actual genetic makeup like PPPP or PP, and the phenotype, which is the observable trait you actually see.
Right. And through his experiments, he figured out that some traits are dominant, meaning they are expressed if the allele is present, and some are recessive, meaning they're only expressed if you have 2 copies of that allele.
This led to his 2 famous laws. The first is the law of segregation,
Which just states that the 2 alleles an individual has for a character separate from each other during the formation of gametes. Each gamete gets only 1.
and his second law.
The law of independent assortment, which says that genes for different characters, like seed color and flower color, assort into gametes independently of each other.
And we use a tool called the Punnett square to do phenotype analysis and predict the probability of offspring inheriting certain traits.
It's a great predictive tool for simple Mendelian genetics.
Of course, biology is almost never that simple. There are lots of exceptions.
Lots. You have incomplete dominance, where the phenotype is a blend. A red flower and a white flower make a pink flower. You have codominance, where both alleles are fully expressed, like an AB blood type.
You also have pleiotropy.
That's when a single gene can influence multiple seemingly unrelated traits. And the opposite is polygenic inheritance, where one trait, like human height or skin color, is influenced by the combined effect of many different genes.
And eventually, all of Mendel's abstract ideas were connected to the physical cell by the chromosome theory of inheritance.
Right. It linked the behavior of his heritable factors to the actual physical behavior of chromosomes during mitosis and meiosis. This also helped explain things like linkage, where genes located close together on the same chromosome tend to be inherited together as a single unit.
Which brings us to the core information pathway of all life, the central dogma, how we get from DNA to a functional protein.
Right. The first step is you have to copy the DNA itself. That's DNA replication. And it's a semi-conservative replication process.
Meaning each new DNA molecule is made of one old strand and one brand new strand.
Exactly. The process kicks off at specific sites called the origin of replication. The main enzyme that builds the new DNA is DNA polymerase.
But there's a problem because the 2 strands are anti-parallel. They run in opposite directions.
Right, so DNA polymerase can only build the new strand in one direction. On one strand, the leading strand, it can just synthesize continuously. But on the other strand, the lagging strand, it has to work backwards, synthesizing it in short, discontinuous pieces called Okazaki fragments.
Which then have to be stitched together.
By another enzyme called DNA ligase. It's the molecular glue.
Okay, so we've copied the DNA. Now we need to get the information out. Step 2 is transcription.
This is the process of copying a gene's DNA sequence into a messenger RNA or mRNA molecule.
Yeah.
The enzyme RNA polymerase binds to a start signal on the DNA called the promoter and makes an RNA copy, stopping when it reaches a terminator sequence.
So now we have the message. The final step is to read that message and build the protein. That's translation.
Right. This happens on the ribosomes. The ribosome reads the mRNA sequence in 3-letter words called codons.
And another type of RNA, tRNA, brings the building blocks.
Exactly. tRNA, or transfer RNA, has an anticodon that matches the mRNA codon, and it carries the corresponding amino acid. The ribosome links these amino acids together one by one.
Translation always starts at the start, codon AUG.
And it continues until the ribosome hits one of 3 stop codons, UAA, UAG, or UGA, which tells it the protein is finished.
In eukaryotes, that mRNA message needs a little processing before it can be translated.
It does. A protective 5-foot cap is added to one end, and a long poly tail is added to the other to help stabilize it. And most importantly, it has to be spliced.
What does that mean?
Eukaryotic genes have non-coding regions called introns scattered throughout them. These have to be cut out, and the actual coding regions, the exons, have to be stitched together. That process is called splicing.
And this process allows for something really amazing, alternative splicing.
Yes. This means that a single gene's transcript can be spliced in different ways. By treating different segments as exons or introns, a cell can produce multiple distinct proteins from just one gene. It hugely expands the coding capacity of the genome.
Our final topic is control. How do cells regulate which genes are turned on or off? This is gene expression.
And this regulation is incredibly complex and vital.
In simpler prokaryotes, they use a system called the operon.
An operon is a cluster of related genes that are all controlled by a single promoter and an on-off switch called the operator.
And there are two classic examples.
The lac operon in E. coli is inducible. It's normally off, but it gets turned on by the presence of lactose. The trip operon is repressible. It's normally on, but it gets turned off by the presence of its end product, tryptophan. It's a very efficient system.
Eukaryotic control is, of course, way more complicated.
Way more. A major level of control is just the physical access to the DNA. The DNA is tightly wound around proteins called histones to form chromatin. How tightly that DNA is packed determines whether the machinery for transcription can even get to the gene.
And these packing patterns can even be inherited.
They can. That's a field called epigenetic inheritance.
What are some other levels of control?
You have proteins called transcription factors that have to bind to the DNA to help recruit or block RNA polymerase. You also have distant DNA sequences called enhancers that can dramatically boost the transcription of a gene.
A really fascinating example of gene regulation is X inactivation.
It is. In female mammals, which have two X chromosomes, one of the two X's in each cell is randomly and permanently shut down early in development, condensing into a silent structure called a bar body.
We're also learning that non-coding RNA plays a huge role in regulation.
A massive role. Small RNA molecules like miRNA and siRNA can trigger a process called RNA interference. They bind to specific mRNA messages and either block their translation or cause them to be destroyed, effectively silencing the gene.
Finally, we have to talk about what happens when the DNA sequence itself changes. A mutation.
A mutation is a permanent change in the DNA sequence. A silent mutation is one where the change of the DNA doesn't actually change the amino acid that's coded for, thanks to the redundancy in the genetic code.
But a missense mutation does change the amino acid.
Right, it substitutes one amino acid for another, which may or may not affect the protein's function. But a nonsense mutation is almost always bad. It changes an amino acid codon into a premature stop codon, cutting the protein short.
And perhaps the most dangerous type is a frameshift mutation.
These are often catastrophic. This happens when you insert or delete nucleotides in a number that's not a multiple of 3. This shifts the entire reading frame for the rest of the gene, scrambling every single codon downstream and usually resulting in a completely useless protein.
And it's important to distinguish between germline and somatic mutations, right?
Absolutely. A germline mutation occurs in a gamete, a sex cell, so it can be passed on to offspring. A somatic mutation occurs in a regular body cell, so it affects that individual, but will not be inherited.
Wow. We have really covered some vast terrain here. From the definition of life itself, through the entire hierarchy, the scientific method, the chemistry of atoms, the 4 macromolecules powering the cell, and the entire flow of genetic information.
And if there is one single takeaway from all of this, it has to be the interconnectedness. Every single concept we covered drives the next one. The chemistry of hydrogen bonds dictates the structure of the double helix.
That structure of the lipid bilayer allows for selective permeability.
And that function allows for the ion gradients that drive ATP synthase. And that energy ultimately powers the variation and reproduction needed for evolutionary adaptation. Seeing it as one big, holistic system, that's the real key to understanding it.
So what does this all mean? We've talked about how complex life evolved. Let's go back to the endosymbiotic theory for a moment. That wild idea that 2 separate prokaryote cells merged to become the first complex eukaryotic cell.
Well, think about the very first step of that process. The ability of one cell to engulf another. What is that? That's endocytosis, a form of bulk transport that we talked about. It relies on the fluid nature of the plasma membrane.
And the reason that merger was so successful, the reason it stuck.
Because the engulfed cell brought a superpower with it. The incredibly efficient pathway of cellular respiration. It provided a massive surplus of ATP for the new combined organism. The entire event was driven by the fundamental concepts of membrane transport and energy efficiency.
So for final thought, investigate the ongoing importance of that volume to surface area ratio. We saw how it physically limits the size of a single cell. Now think about how large multicellular organisms have solved that problem.
They've evolved complex structures, folds, tubes, branches, all designed to do one thing: maximize surface area for exchange. Think about the alveoli in your lungs, the villi in your intestines, the veins in a leaf. It's all the same solution to the same fundamental physical constraint, a constraint that is still shaping the architecture of every living thing on this planet.